Hi, everyone. Thanks for being here, and thanks so much to the organizers for the opportunity to talk about our multi-omic work on colorectal tissues from people with and without long COVID. I also want to acknowledge all the people who've worked on this particular project and on all of our long COVID projects. These are all huge group efforts. So we and others have previously shown that SARS-CoV-2 can cause chronic immune activation and dysfunction and can persist at low levels in gut tissue years after initial infection. These images that Tim also showed are from our STM manuscript, where we identified cellular SARS-CoV-2 RNA in rectosigmoid lamina-propria tissue using RNA-scope in situ hybridization in a subset of participants studied for up to 2 years after initial infection. However, little is still known about how and how much viral persistence and immune dysfunction may affect overall immune function in the gut in people living with long COVID, especially in a large, well-characterized cohort. And so to that end, in an effort to answer these questions, we worked with colorectal specimens collected through the LINK cohort, headed by Dr. Michael Peluso. We analyzed colorectal tissue from 56 participants. 43 of these participants had long COVID, and so those are termed LC samples, and 13 had acute COVID infection but recovered fully, and those are fully recovered or FR samples. And as you can see from this table, there's no significant difference between age, sex, or time since initial infection between our long COVID and recovered groups. And so these are all the strategies we employed. We performed quantitative PCR on RNA extracted from bulk tissue using an assay that targets the spike and nucleocapsid genes. We performed duplex RNA scope in situ hybridization on FFPE tissues. We used end-counter analysis systems to profile the expression of 750 human immune response-related genes, as well as 33 custom add-on genes, including SARS-CoV-2 and EBV viral genes on all of our samples. And finally, we performed 10x xenium digital spatial profiling on FFPE tissue using a pan-tissue panel, as well as unbiased RNA-seq, MNGS sequencing, and single-cell RNA-seq. So qPCR results were largely negative. Out of the 56 samples tested, only one was positive for spike RNA. But previous work has shown that in situ hybridization can actually be a more sensitive method for detecting low-level viral transcripts in tissue. And so with that knowledge, we implemented an RNA scope assay with a duplex cocktail of 2 probes targeting both the spike and ORF1ab regions of the SARS genome. The spike probe is on a green chromogen and binds to single-stranded RNA, and the ORF1ab probe is on a red chromogen and is a sense probe, meaning it targets the antisense or double-stranded ORF1ab strand that's typically only present during viral replication. And we validated the assay using a SARS-CoV-2 infected mouse model. That's what you're looking at here. And each sample was also run with a pre-pandemic colorectal tissue section on the same slide to assess the noise level of the SARS probes, which, as you can see on the left here, is quite minimal, as well as a human housekeeping cocktail on the right to confirm assay performance and RNA integrity. We detected SARS single-stranded RNA, spike RNA, in 10 of the 56 samples analyzed from this cohort, and double-stranded RNA in 2 out of those 10. The 2 samples that did exhibit double-stranded RNA were an average of 172.5 days from initial infection, which was also their most recent infection, while the single-stranded samples were an average of 362 days from most recent infection, and 613.6 from initial infection. We did not detect RNA in any of the fully recovered individuals. The RNA that we see in these samples could be in a variety of forms, ranging from intact, partially or heavily degraded, or to some extent low-level replication competent. Co-localization of the red and green signal, like we see on the top left section, could be indicative of replication competent virus, but it's also possible that we're seeing fragments of partial genome transcription in cells harboring defective viral RNA, like we see in other chronic infections. And so this leads to our host transcriptomic analysis. This data is from our nCounter assay, where we got transcriptomic data from a panel of 750 host immune-related genes and 33 custom viral genes. And after normalization using housekeeping genes and positive and negative controls, our analysis revealed several significantly differentially expressed genes related to various aspects of immune function between our long COVID and fully recovered samples. On the top, the blue boxes are long COVID samples, and the orange are fully recovered. And on the left, the green section is genes upregulated in long COVID, and the purple is downregulated genes in long COVID. And I've also annotated the RNA-scope positive samples with red asterisks on the top. You can see that the fully recovered samples cluster together there in the middle, and we see that the long COVID samples seem to cluster into two groups. One on the left that more so displays downregulation of immune clearance-related genes, and one on the right that has an upregulated inflammatory signature. And so this is just another way of looking at this data on a volcano plot. Again, on the right, we do see upregulation in long COVID of genes related to chronic immune activation and inflammation, but the most significant differences are on the left, where we see significant downregulation in long COVID of various genes related to immune trafficking, immune signaling, pathogen recognition, antigen presentation, et cetera. And so this is another heat map, a clustered heat map from our bulk RNA-seq data of all the genes that are significantly differentially expressed in our long COVID versus fully recovered comparisons. The rows in this heat map represent genes, while the columns represent the individual samples. And at the top, you can see the distribution of samples from different long COVID clinical phenotypes. And we see that only the fully recovered phenotype in that salmon color on the bottom row displays strong clustering, whereas samples from the other conditions are distributed throughout the heat map. And so one of the main questions we wanted to try and answer is, well, what cells are driving the significant dysregulation of immune function, especially the stark downregulation of various immune function genes that we see in the nCounter data. So taking a closer look at our single cell data, these UMAPs show that the frequency of the cell type distribution is similar between the fully recovered and long COVID data, but I'll talk briefly about two specific clusters, the macrophages and the plasma cell cluster. So looking at the macrophages first, this is a gene ontology bar plot from our gene enrichment analysis, and we're seeing significant differences in several immune pathways comparing long COVID to fully recovered. As you can see on the bar plot, but this is just showing the magnitude of the change, not the directionality. But we can gauge that from looking at this heat map, where we have genes on the bottom and pathways on the left. And we see that most of these are red or upregulated. But it's also important to consider that the overall abundance of macrophages in the gut is quite low compared to other immune cells. So next we looked at the GO analysis for the plasma cell clusters, which are much more abundant in the gut. And we see a similar pattern in the gene ontology of significantly different pathways when comparing long COVID to fully recovered, including things like Fc receptor signaling, lymphocyte regulatory pathways. And then looking at the heat map, we see a lot of blue on this one, or downregulation of these pathways that would mediate adaptive innate immune responses to viral infection, which may explain why we're seeing significant downregulation across many of the immune genes in our nCounter data. And I'll just touch on this briefly. This is our Xenium digital spatial data that is kind of the last piece of this project that we're putting together. This is the output. We have 4 long COVID samples and 2 fully recovered samples that we're working on analyzing, and much of this is being done by David Mason, a former postdoc and colleague of ours. And so to summarize our findings, we demonstrated that SARS-CoV-2 can persist at low levels in the gut in some, but not all long COVID cases. We actually think that our positive count is a slight underestimate because of how low the overall SARS RNA burden is per section and how thick the tissues we're analyzing actually are. Each section that we stain is only 5 microns thick, but the FFPE blocks containing the bulk tissue are hundreds of microns, so it's possible that we're missing some. Downregulation of certain genes associated with immune-mediated viral clearance and cytotoxicity suggests a mechanism for persistence of SARS RNA in gut tissue, but our transcriptomic analyses also demonstrate that long COVID is associated with broader tissue inflammation. Our single-cell data indicates that immune cells are responding in a discoordinated fashion in the gut. And finally, but also very importantly, these findings appear to be specific to gut, which we know is an immunoregulatory environment. Like Tim mentioned, we don't see this pattern of differentially expressed genes or viral persistence in peripheral whole blood samples from the link cohort that we analyzed with the same assay. Yeah, so the take-home message, like Tim, I want to leave you with is that long COVID is a tissue-based disease, and it will be critical that this is taken into account during biomarker development and clinical trial design. And with that, I want to thank all of our lab members and collaborators, as well as our funders, and most importantly, I want to say a huge thank you to all of our study participants, and thank you for your attention.